So in this video, I'd like to talk about transcription, which is the process by which RNA strands are made. So we learned earlier about our two twos, that a DNA strand has an informational part and a structural part, and that it has two ends, 5' end and a 3' end, and our four rules. So while we didn't emphasize it, these rules and these two twos apply both to DNA and to RNA. And there's actually, among these rules, there's only this one difference, which is where DNAs have T's, RNAs have U's. So let's see how that plays out by looking at the actual structure. So here is a 4-base single strand of DNA. So if this were an RNA, how would it be different? Well, it would be different in two ways. The first we said is that instead of having a thymine, it would have a uracil. And you can see that thymine and uracil are very similar compounds. They differ only at this one position. And the other is that whereas the DNA would have a hydrogen at each of our bases along the backbone, that RNA has an OH group. It's a hydroxyl group. And this is actually why we call DNA deoxyribose. So ribose is where we get RNA. And deoxyribose means deoxy less oxygen. So instead of having this OH as does ribose, we have just an H in DNA. Okay, so then can we make some sense of why there are these two annoying and hard to memorize differences? So first let's start with uracil versus thymine. So note that uracil is very similar to cytosine, to the C base. So let's think about that. That means if we had uracil in DNA, that it might be easier to mistake uracil for cytosine and therefore to place the wrong base, right? If we had a cytosine in there, we might mistake it for a uracil and say, oh, that should go with an A. If we had a uracil there, we might mistake it for cytosine and go for a G. In particular, ultraviolet light deaminates cytosine, so it takes this, it's a kind of damage that takes this NH2 group and turns it into an oxygen. So that means actually that ultraviolet light directly converts cytosine to uracil. So if we used uracil in our DNA, we would have this constant process by which our cytosines would be getting turned into uracils. So we'd have a lot of mistakes. We'd have a lot of mutations. Okay, so that's an argument for why we don't have uracil in DNA, but then why don't we just have thymine in RNA? Why use different nucleotides in the different molecules? Well, uracil is less energetically costly to actually produce, because remember, every time we have a uracil in a DNA and in an RNA, it has to be produced. Every time we have a thymine in a DNA, it has to be produced. These aren't free. The cell has to produce these things. Uracil is less costly, so RNA, which is hyperabundant in the cell, it makes sense that for RNA, it'd be preferable to use the lower energetically costly base. So uracil is the less costly one, which is put in the temporary abundant RNAs. Thymine is the more stable one, the one that looks less like cytosine, and that therefore it's put in the DNAs where mistakes are more important and more costly. Okay, what about the backbone? Well, RNA actually, in addition to the classic role where it just tells us which proteins to make, which we'll be getting to, other RNA molecules function by folding into three-dimensional structures. We'll talk about that soon. But it means RNAs function in part by folding up into three-dimensional structures. So this OH group makes the backbone of RNA more flexible than it would be if it were a DNA. And so this backbone, the RNA backbone, is more flexible, and that allows it to function in ways that single-stranded DNA could not. And what about DNA? Well, for DNA, the lack of the OH group makes the backbone more rigid, opposite of what we just said. So the DNA double helix, this makes the DNA just needs to be stable. It doesn't need to fold up. It doesn't need to do tricks. It just needs to be stable. So having a DNA double helix makes it more stable than if we had an RNA double helix. So it means it's less prone to breaks, mutations, and so forth. Okay, so uracil and thymine is this sort of trade-off between similarity to cytosine and energetic costliness. And OH versus H is this trade-off between flexibility and stability. But the main point here is just to point out that RNA is almost exactly the same as DNA, and it just has these two slight differences. Okay, so just like DNA strands, RNA strands are created by complementing one strand of a DNA in a process called transcription. So let's see what that looks like. So we still have these pairing rules, except for now, when there's an A, we're going to put a U. When there's a C, we're going to put a G. When there's a G, we're going to put a C. And when there's a T in the DNA, we're going to put an A. Okay, so very similar to the rules, again with just these two differences. Okay, so we learned that for DNA replication, that when we create new DNA strands, that what we do is we separate the two DNAs, and then base by base, we create these new strands. I'm sorry, for some reason this is a little bit slow. So base by base, we create our bottom strand, and then base by base, we create our top strand. I guess it's more than a little slow. Okay, so what about for transcription? Okay, so the main point here is to realize that with DNA replication, the point is to duplicate the whole DNA. Remember, when we replicate DNA, what we're trying to make is two identical full copies of our initial DNA so that our descendant cells can each get a copy. For transcription, it's a different point, right? There's a different function. We're not trying to make a copy of the DNA. That's not the point. The point is to make an RNA, is to get a bit of RNA that will give us the instructions for making a machine. So usually that RNA will then be translated into protein. We'll talk about that in the next video. And then sometimes an RNA, actually, it already will have its machine properties, which we'll discuss. But what that means then is that we don't want to just make a full copy of our DNA when we do replication. We want to have some specific sequence, some specific part of the RNA. And that the part that we want is the part that contains the message that has some function. So here I've done tag a cat. Sorry, I forgot to, I meant to mock up something with a graffiti tag on a cat. But so tag a cat, if that's the, in this case, English 3-word message that we need, then let's think about how we would make that. Okay, so if we want to have an RNA that just has tag a cat, then we would separate our two strands, and then we would add our nucleotides one at a time. Sorry about that. I don't know why this is so slow, but it goes faster when I do it this way. Okay, so what we want to make is just tag a cat. So notice, well, first thing, let me point out. So this is RNA, and I just told you RNA doesn't have T's, it has U's. So what am I talking about here? Well, I couldn't come up with any good English language words that only used A, C, G, and U. So instead I cheated. I put T's in our RNA. Okay, so but you're right, there shouldn't be T's there. Okay, but if I wanted to make this tag a cat, then I would just do these 7 bases. Notice I don't want to do the stuff right before tag a cat. I don't want to do the stuff right after tag a cat. The other thing is, so do I want to do both strands? Okay, so we can do the both strands for the same region, and then it's atgulgikutuga, which doesn't mean anything in our language, right? In the language of English that we're using here. And so if we want this message, if this message is the functional RNA, then we're just going to do this message. We're not going to do extra stuff. We're not going to do the whole DNA strand, and we're not going to do the opposite one. Right, so just like the reverse strand of tag a cat is nonsense, the reverse strand of our useful functional RNAs that the cell creates are usually nonsense. Okay, but then what are the parts that are useful? Well, the parts that are useful are the transcribed regions, which we learned are our genes. So let's see what that looks like for our double-stranded DNA. So that would mean that we have some region that gets transcribed, and then that RNA goes off and becomes its own independent RNA molecule. And the other point is to recognize that we don't just need to make one copy of this, we can make many copies. So again, it's not unlike DNA replication where we're trying to make exactly one other copy. Here with RNA, we're going to make as many copies as the more protein that the cell is going to make, the more RNAs we'll make. Okay, so what determines where transcription starts? Well, it's the presence of a promoter. So a promoter is just this sequence that has the chemical properties that attracts the replication machinery, the DNA polymerase, which directs the DNA polymerase as to where to start transcription. So the other thing, so if we were to look at a region of a chromosome, a region of a double-stranded DNA, what we would see is that there will be within it, not the whole thing, but only little parts where the DNA strands have been separated and the RNA polymerase is creating RNAs. So on the left, we've got one, and then in the center, we've got another gene where things are being transcribed. Okay, well, what about the third one? Which strand is going to be transcribed? So it's sort of natural to think maybe the same strand, this strand here that's connected to the bottom, maybe that's going to be the one that's transcribed. But it turns out that the way to think about it is sort of independent, that like this one, something's happening here, and then this one, something's happening here, and they're not really talking to each other, and they're not really related. So, and which one is the bottom and the top, if you think about it, that's kind of just depends on how we draw it, because we could flip this over. Remember, this is a double-stranded DNA chemical molecule that exists, that's diffusing through the cell, that's flipping all around. So when we draw it in one way, that's just because as humans it helps us understand it. But there's nothing special. This bottom strand is not fundamentally different from the top. So that could mean that, you know, over here, if we rotated this thing all the way around, then this would be the bottom strand, and then this gene would be the one that's doing it the way we expect, and these ones would be doing it upside down. So our upside down, right side up thing is just kind of our natural way of thinking about things, but in fact, genes can be transcribed from either strand. It just depends on which strand the message is. Maybe tag a cat is down here, but gag a tat is over here, and yikes, I should have planned this out. AT is over here on the top. Okay, so main point there being that for a given gene, only one strand is transcribed, and that strand can be different between different genes. Okay, so the take-home points here. RNAs are created by a process called transcription. RNA is typically only transcribed from one strand for a given gene. Transcribed regions are called genes. On a chromosome, sometimes one strand is transcribed, but in other genes, sometimes the gene is transcribed. It's different for different genes. And what determines the start position for transcription is the promoter.